So welcome everyone. It's great to see such great numbers. My name is Vanja Anderle. So let's first maybe go just check the agenda. And to quickly explain, so in parallel to all the talks you see here, we will be having a Q&A sessions. It will be a session done through the Q&A app in Zoom. So below in your control panel, there's a button, Q&A, where you can post questions at any time during the webinar. Yes, so now without further ado, I would like to welcome the CEO of Cosylab, Dr. Mark Pleško, to say a few words. Welcome, Dr. Pleško.
Welcome, everybody. I thank over 450 participants for attending Cosylab's first seminar about BNCT. This is the first seminar in the series which will be organized by Cosylab this year. And I think it's a great turnout, and it shows that BNCT is really gaining traction. We are very, very proud to have so many esteemed speakers from around the world. They will share their views and experience about BNCT with the broader BNCT community, and of course with everybody else who is interested in this topic. And we really thank you from our heart for devoting your time to this important topic. Now, as you know, BNCT has been around since the last century, but it has really started picking up the steam over the last few years due to recent advancement in accelerator technology and hopefully also in the future drug technology. So thank you very much for being here. And I will now like to invite Jay Flanz, PhD, to start with the sessions. Jay is president and chair of the Particle Therapy Cooperative Group, PTCOG. He is retired associate professor at the Harvard Medical School, and he's retired technical director at the Burr Proton Therapy Center of the GmbH, Massachusetts General Hospital. Thank you very much, Jay, and I hope everybody will enjoy this webinar. Thank you.
Okay. Hi, it's also my pleasure to welcome you to this BNCT webinar. I'm Jay Flanz, chair of PTCOG. That's the Particle Therapy Cooperative Group. One of our missions is to promote global activities in particle therapy with the goal of improving the treatment of cancer to the highest possible standard. There are many aspects of radiation therapy. One of the reasons that radiotherapy can work is if the target tissue and the healthy tissue are differentiated. That could be from targeting, or it could be from modifying the relative sensitivities of the cells. One of the BNCT pioneers, William Sweet, who was a neurosurgeon at MGH, designed treatments in the 1950s. And during my time working at MIT, MGH, and Harvard, that name was actually used on both sides of the BNCT coin. Hatanaka, a student of Sweet, treated patients in Japan, and they had nice results for GBMs. But BNCT is a highly interdisciplinary activity. It requires technology, physics, engineering, biology, chemistry, and oh yes, it requires medicine. Getting all of these pieces together has been a challenge. A few years ago, some people expressed concern to me about the lack of presence of BNCT in PTCOG. It is, after all, a particle radiotherapy modality. My response at the time was, BNCT reports are welcome. We just need people to submit abstracts. Even today, for the PTCOG 60 that will happen in Miami in June, as of February 17th, we had 4 BNCT abstracts, and as of February 25th, it was 5 out of 430. There has been much work on the technology, identifying even building applicable accelerators and neutron moderators for reactors, but the whole package hasn't been very visible recently. 10 years ago, I believe BNCT was on the 3rd generation of boron delivery agents in tumor targeting molecules. One time when I visited MIT some years ago, they were using it for arthritis in dogs. But now there's growing clinical work in Japan, a growing revival of interest in the US and the rest of the world as well. I even see that Bill and Ted are starting another excellent adventure. And hopefully more of you will understand that reference after the webinar. Anyway, it seems now is a time appropriate for this webinar to review where the field is, where it needs to develop, or how it might be marketed if it's believed it's ready for broader clinical use. I'd like to encourage you to join the PTCOG Bnct subcommittee, and I look forward to hearing your discussions and to see this modality have a larger presence in the upcoming PTCOG conferences. So I really do hope that everyone is going to enjoy this meeting, and it's great to know that it's the first of a series. Excellent speakers have been invited to this webinar. So to start, it is my pleasure to introduce Paul Busse. Professor Dr. Busse is the MGH Clinical Director of Radiation Oncology, and he's the incumbent of the Joseph Cotchett Endowed Chair in Radiation Oncology. This was the first MGH endowed chair for the department. Back in 1998, Paul was quoted in the MIT News saying, "The promise of BNCT is the selective irradiation of tumor cells in a way that is not achievable using current forms of conventional radiation." So far, our expectations have been met with respect to the scientific goals of the clinical trials. Paul was the principal investigator for the program grant of BNCT at MGH and MIT. And in 2013, he was on the panel of the NCI Radiation Research Program Meeting Report, Clinical Research in Neutron Capture Therapy. You can read about some of the history in Paul's chapter in the book Frontiers in Neutron Capture Therapy. Actually, Paul and I share some similar backgrounds. His family tree consists of a long lineage of florists, and my name is Flanz, coming from the German word meaning kingdom of plants. So I guess Paul and I share some common roots. We worked together at MGH, and though I admit in the times that I've been drinking with him, he didn't mention BNCT very often, this subject has clearly been an integral part of Paul's career. So it's good to see you, Paul, virtually. Thank you for participating in this meeting. And I really hope that everyone enjoys the meeting. Thank you.
Well, thank you, Jay, for the kind words. You know, it's been a great working with Jay for a number of years at MGH, and he's been a very important part of our department and the Proton Center. I was looking at this group, and you know, there are very few people in the world who have treated patients with every subatomic particle, neutrons, protons, electrons. Wolfgang and I are two of those people, and Jay Flanz is someone that's produced all those particles. So you joined the club, Jay, and we wouldn't be able to do what we do without you. Okay. So my task today is to discuss the history and the early progress of BNCT, but also I've taken an opportunity to digress even further and to sort of frame the need for BNCT and why it even exists with the full spectrum of radiation capabilities that we have now, which are nothing like we had even 10 to 20 years ago. And so as part of that, I think it's important to say it really begins with an understanding of the limitations of conventional radiation. And this audience is very diverse. And so for some of you folks, this is very elementary and redundant. But for others, I think it really will serve to frame why people are interested in BNCT, where something that's been around for so long. So there are two ways to treat patients with radiation now. One, photons, and the other one was protons. The most common type is with proton radiation, and it is the use of high energy linear accelerators, 6 to 35 million volts. We also use cobalt, which is a lower energy. We use superficial electrons produced by these accelerators and various isotopes for brachytherapy. So all of these sources, although they're different in their source, they have something in common. And that is they interact with matter through electrons, and that there's a limitation by depth and complexity of a tumor target. And unwanted radiation, both proximal and distal to a clinical target, receives radiation, sometimes more than the target. And there's no discrimination within the target if the target involves normal structures between the tumor and the normal tissue. So we've made the most of this over the years and done some remarkable work in our ability to target, but biologically, it remains these same elements of the problem. Proton radiation is something where J. Flanz has made his mark in the world. And this is a more precise way of delivering radiation. It is a charged particle, a stripped hydrogen nucleus with a positive charge. And this has a very sharp beam edge. And it also has this attribute of going into tissue for a certain depth, which is limited by the energy, and then depositing most of its energy in this characteristic Bragg peak. These particles are produced in a cyclotron or a synchrotron. They're high energy. It's complex, it's expensive, and it's a limited resource, even though there are many more proton facilities being produced and is in service compared to when I started as a resident. The only place was either Loma Linda or MIT or the Harvard cyclotron across the river. There are many worldwide, and for a number of disease sites, it's very important. However, if you look at the percent of radiation dose as a function of depth, and depth in water is the same as depth in tissue. If this is the depth dose profile for photon radiation, and this is the depth profile for a spread out Bragg peak for proton radiation, the green shows unwanted X-rays, both at the beginning, at the entrance point, and at the point distal to the clinical target. So for the same target, you get less dose with protons than with X radiation. But that's far from ideal. If this is another representation of the photon depth chart in an idealized proton depth chart for a small clinical volume, you can see that compared to the ideal, which is this, even protons have unwanted radiation. And there's only one way to deliver radiation with a distribution of dose, which looks something like this, and that is to irradiate something from the inside out. And that's the promise of BNCT, which is a biological targeting and delivery of the radiation. Now, this is achieved by the BNCT reaction, and you'll probably see this a couple other times during this talk, but essentially a low energy neutron is then selectively taken up by boron 10, and it is taken up by other atoms as well. But within a very short period of time, the boron 10 nucleus disintegrates into a lithium 7 nucleus and an alpha particle. Now, what's notable is the very small distances that these particles traverse before they give up a tremendous amount of kinetic energy. And then that is deposited where we want, ideally within the cell nucleus or within the range of macromolecules to produce enough destruction so that the cells are killed basically from the inside out. We measure the efficiency of radiation in something called RBE, and the RBE is defined for orthovoltage X-rays as 1. The RBE for fast neutrons is 3, and the RBE for most boron 10 reactions is 3.8. So you can see there's an advantage to this over straight X-ray. So not only is the radiation targeted, but it also has a higher biological effect. So ratios, we also have to have enough boron 10 in the target tissue to be effective. And so ratios of 3.5 and 4 to 4 have been measured in many human tumors, both by just direct measurements as well as by looking at radionuclide measurements of a boron carrier. And the normal tissue, to a first approximation, follows the time course and the concentration of blood. So I'm going to leave this more to the physicists, but basically there are 3 neutron energy ranges. Thermal neutrons are the slow energy that actually produce the interaction with boron 10. Epithermal neutrons are within a higher energy range, and we need these for the delivery of the radiation to deeper targets within tissue. Fast neutrons are a totally different beast, used for therapy and may have some use for BNCT, but that's not the primary source that we're talking about. Again, neutron sources, like proton sources, are limited, in which case, up until only recently, they have been extremely restricted because they've been reactor-based. And research reactors are located away from medical facilities. They're not designed for clinical work or patient care, although my colleagues at MIT did the very, very best to make that reactor patient-friendly and clinical-friendly. And even the MIT reactor, compared to many, is very, very close to medical facilities. I can look out my window right now and almost see the smokestack from the nuclear reactor lab, but still it's not the same as having a treatment facility within the physical plant of a hospital. So there is no clear path for a small hospital-based system. Professor Harling used to say, well, they're all small portable uranium-based reactors that could be used, but I can tell you that is never going to happen in a hospital. There are also tiresome issues like uranium disposal and holding on to that for the next 500 years. So reactor bases really have been the source of our clinical work so far, but they're not the future. Now, at the time, BNCT was first conceptualized by Locker and others, that therapeutic radiation was even in a more primitive state than it is now. There was low energy orthovoltage X-rays. The dose was 100% to the skin, terrible outcomes, terrible complications. This was pretty much for all disease sites. So really the time was ripe for thinking about radiation in a different way. Bill Sweet was a neurosurgeon at MGH. He recognized the futility of conventional radiation for his patients. And he collaborated with Gordon Brunel at MIT at the time of construction of the research reactor to install a site for clinical use for neutrons. So when this reactor was built, there was a clinical suite on the ground floor beneath the actual reactor itself. And a beam came from the ceiling. It was all tiled. It had an operating room table. It was sterilized, and it was actually used as an operating suite. And the first patients were treated from 1951 to 1960. Now, the first clinical trials that Brunel and Sweet conceptualized and put together were not done at MIT because they weren't ready, but they were done at the Brookhaven National Lab. There were glioma patients. There was no real enrichment for boron-10. It was sodium tetraborate. There were thermal neutrons with very limited penetration. So there was no evidence of tumor response, and there was some toxicity. That's a misprint. So in 1960, there was a second generation of trials at Brookhaven National Lab with some crude dose measurements to try and quantify how much radiation was given, but there was severe scalp injury and no tumor response. Third BNCT trial was initiated at MIT in the new operating suite. 18 patients resected as much as possible, in which case there was an IV injection of a boronic compound, which theoretically had some greater targeting ability. The scalp and the dura were reflected to improve the penetration of the thermal neutrons out of the MIT beam, but there was no measurement of tumor or normal tissue dose, no improvement in survival, and postmortem exam revealed severe parenchymal injury. And one of my colleagues, who was a medical house staff at the time at MGH, said that these patients would come back from MIT and go straight to the emergency room at the MGH, and they were in pretty tough shape after this. So this was where the clinical trials ended. There's no quantification. The qualities of the beam were not optimal. Patients had to have open craniotomy. Complications were severe, and so the trials ended in the US. As Jay mentioned, Hatanaka was a research fellow and a protege under Bill Sweet, and developed a new compound, BSH, which is a polyhedral boron, which contained a number of boron atoms. It had a high concentration of boron-10 per molecule, and it was definitely improvement over any of the compounds used previously. So there are a number of clinical experiences in Japan run by Hatanaka and colleagues for glial tumors. And the initial experience was very encouraging, close to double the median survival, but there was a lot of case selectivity in those patients. Mishima is another name to know, shifted the interest from GBMs to melanoma, developed BPA, which is a boronated phenylalanine, which is a boron 10 carrier that had a little bit different approach. And this was seen as designed as a metabolic precursor to melanin, so that this would be incorporated into the macromolecule of melanin and have a selective uptake in melanoma cells. It was water soluble, particularly when conjugated with fructose. We used the L-diastereomere isomer, and it had a 3 to 4 tumor to normal tissue. So refinement of BNCT in Japan, Nakagawa has done a tremendous amount of work, as well as many others that preceded or followed him. They treated primarily glial tumors, grade 3 and GBM, used BSH as their boron 10 agent, used open craniotomies to improve neutron penetration. Again, used reactor-based sources. So we're doing craniotomies in patients away from the hospital. Close to 200 patients were treated on numerous protocols. Anecdotal successes. And it's difficult to know exactly what the true line for the successes were, because they used, sometimes they had standard radiation, sometimes it was just BNCT, different ways of generating dosimetry, combinations of compounds. But I think the distillation of it all was that there was something there, but it really wasn't defined in a way that we all could say this is the next best thing. So there was a resurgence of interest in the US and Europe based on the initial experiences in Japan. So there was a combined European effort led by Wolfgang with radiation at the Petten Research Reactor through the EORTC. And there were two groups in the United States, MIT and Tufts, and later at Harvard, that I was fortunate enough to participate in. And then also at the Brookhaven National Lab. We have two compounds that we use in clinical trials, which is BPA, which I mentioned, as well as BSH. And the Harvard MIT program, we had clinical trials with intracranial tumors, glioblastoma, metastatic melanoma, and a phase 1 CNS toxicity trial. We also had a peripheral melanomas at the extremities. Also, that was a phase 1 trial for dermal toxicity. These were all NIH and Department of Energy funded. They're peer reviewed and IRB approved. Now, our initial clinical trial had 24 subjects, 22 were radiated, median age of 56, GBMs, 2 with metastatic melanoma, and we went through a number of dose ranges looking for the maximum tolerated dose. We also increased our drug concentration of BPA from 250 milligram per kilogram to 350 milligram per kilogram, and did all this successfully with a modicum of toxicity. This is a great slide. This shows the where our initial radiations were done at the MIT reactor. So we treated these patients in the same room, the same operating suite as William Sweet did. This is an OR table on a hydraulic lift. The beam came out of the ceiling, and the patients had to be almost immediately touching the ceiling to get the best penetration and dose. We monitored patients through a view window. We monitored pulse oximetry as well as EKG, and the radiations took a long time. So it's very uncomfortable for everybody. It was uncomfortable for the patient. It was uncomfortable for us watching them and trying to get them through. This is a view from this is the patient's head looking from the vertex, and you can see how close the field is to the beam coming out of the ceiling. One thing we found was this is what we want to see in radiation oncology. We look at dose volume histograms from normal tissue and a clinical target. We want to see as wide a separation as possible. And one of the things that BNCT has allowed us to do is to look at the dose in the normal brain versus the dose in the clinical target. And you can see a wide separation. So you know there's something to this. We also worked out the pharmacokinetics of BPA after an IV infusion, and it's a two-component system. After the initial infusion, a rapid redistribution phase, and a smaller rate constant for renal excretion. So we could use these, and it's a very consistent model. So you don't have to take time points, every 15 minutes through an infusion. Just to show you some anecdotal cases, this is a radiograph of an occipital melanoma before neutron capture therapy. And I can tell you with complete certainty that this was treated with conventional radiation, that you would not see this result. This is the occipital melanoma with a rim of edema around it. And you can see it on the sagittal view here. This is 6 months following neutron capture therapy. And I can assure you this is the same patient. And the lesion is completely gone on both views. And it stayed that way until he developed the metastatic disease elsewhere. This is another example of a response following neutron capture therapy. And this is an enhancing lesion of a good-sized glioblastoma with the enhancing ring, both on the MRI and on the pretreatment CT scan. And this is the CT scan 4 months after neutron therapy, which the important thing is, it's not surprising the necrotic center is still there, but there's complete loss of this enhancing ring, which is the viable aspect of the tumor. This is an interesting patient. This is a woman that had a glioblastoma right here in the temporal lobe that we treated. And then 12 months afterwards, the tumor is completely gone at this site, but she developed a second glioblastoma on the contralateral part of the brain. So we treated this with 60 gray of conventional radiation, this tumor volume, and followed her afterwards with serial scans. And the site of the first GBM remained free of disease. So I find this figure interesting because we measured the volumes of the tumor after irradiation. And after neutron capture therapy, you can see with successive scans, the volume continued to decrease and stayed completely gone for 12 months after treatment. But the same tumor volume, actually a smaller tumor volume, after conventional fractionated high dose radiation just continued to grow. So there is definitely something to the BNCT reaction that is completely absent from photon radiation, even to the same calculated dose. In our phase 1 trial, as you know, the outcome is tolerance. However, we're all looking for some benefit if it's there. So our median survival is 12 months, which is what the standard is for resected post-op treatment and patient treated with post-op radiation therapy for glioblastoma. So in some ways, our unresected patients treated with one fraction of BNCT was no worse than the standard of treatment. So as I mentioned, we had some complete responses. We did have volume measurements, which showed encouraging radiographic responses. 13 out of 17 evaluable patients showed a response, and most pronounced in smaller tumors. Now, at the same time, we were conducting our work at MIT. The other group that was really doing very well was a multinational prospective cooperative effort led by Dr. Sauerwein. He ran these through the EORTC, which is a cooperative group, which meant that there was one extra level of scrutiny and paperwork that he had to do, but it would run like a regular cooperative therapeutic trial. So there was a phase 1 trial for glioblastomas for complete or subtotal resection with BSH. They did a very detailed evaluation of radiographic and cognitive changes after BNCT, which is the endpoint of a phase 1 trial. And they also did an EORTC trial looking at the biodistribution of BSH and BPA, which led to some very, very good detail on what to expect for where the boron 10 goes in these tumors. Probably the most promising results to date have been BNCT for recurrent head and neck cancers. And since I treat and have treated primarily head and neck cancers at Mass General for the last 20 years, and even at the Dana-Farber before that, this is a very, very difficult problem. There are very few things we can do for these patients, particularly if they've had previous radiation. Responses are poor, survival is poor, and there are very few good options. But there are very careful studies, particularly by the Finn group in Helsinki, and one well-reported study. They had 30 patients, and 43% had a complete response, and 30% had a partial response, which is remarkable. And a median survival of 13 months, and primary site control of 8 months. And some of these patients even had controls at 2 and 4 years later, which is truly remarkable. And I think could be one of the more promising sites for BNCT. And there are plenty of recurrent head and neck cancers. This is an example that was published in the Red Journal, which is a recurrent cancer right here. And you can see this patient after treatment, and this is clearly gone. So my last few slides, I would be remiss if I didn't pay due diligence to Otto Harling and the many colleagues at MIT that developed a fission converter beam. Otto was quick to recognize the limitations of the beam that came out of the ceiling used 50 years ago by Dr. Sweet. And we knew that we had to trim off the thermal contamination as well as any fast neutron contamination, and have a more collimated epithermal beam with a higher flux. So he developed this fission converter beam with some spent panels of uranium in the wall of the research reactor, and then developed collimations and moderations of the beam so that it came out in what looked like a clinical snout, and with a very good profile. And so the FCB epithermal profile is sort of the gold standard, if you will, for accelerated based neutron. So this is a door into the treatment room. This is a control monitor. We could watch patients. We had a port to measure blood, and it was a very great experience. And the last slide, if you look at the dose volume histogram of single field, this is the normal brain. This is the beam we originally used, and this is the fission converter beam. You can see even a wider separation between the target and normal tissue with the appropriate neutron source. So I will end there. And I will turn this over to my longstanding colleague, friend, and counterpart, Professor Wolfgang Sauerwein.
Thank you, Paul, for this kind introduction. I will immediately continue where you stopped, but coming from a very different angle, even if I'm deliberately selected some slides you already have shown. So I'm looking now as a retired professor of what I made in the last 30 years. It was a lot of science. We wrote a lot of papers. We made a lot of experiments. We had very nice designed clinical trials, but we are far away from any routine clinical practice. So in the limited lifetime I still have, I thought I should make efforts to bring BNCT really close or in routine clinical practice. Why is BNCT important for the further development of radiotherapy? It will never replace conventional radiotherapy. It adds something. Why do we need it? And what do we have to do to implement it? That's a question I would like to try to answer. Paul coming from a different point of view, but being in the same spirit than myself, has already shown these inherent challenges with current radiotherapy techniques. And I always like to say, between this image and this, there is a big distance in years, but the principle is exactly the same. We always treat a volume of tissue. Paul already insisted, so I would not like to lose a long time in this slide, but we have inside of the target volume normal tissues that will be destroyed. We have outside normal tissues that will receive a biological important dose. Our image guidance techniques are far to be perfect. And at the end of the day, it's always a physician who has to define the target volume, which means from one physician to another, the target volume will be slightly or even considerably different. And the physicist will make all efforts to precisely deliver the dose that's needed to this target volume, but there is a huge unprecision prior. So all current radiotherapy techniques, the most modern one, have this problem. And this implies that unless some entirely new way of delivering radiotherapy is developed, any improvements in the efficacy of radiotherapy will be incremental rather than substantial. Bnct is different. I call Bnct a vectorized external beam radiotherapy, which is targeting single tumor cells. And it offers us the possibility for the first time not to irradiate a volume, but really to irradiate the disease. I like this little thing to show these ugly gray tumor cells loaded with boron 10. We make this reaction with the neutrons and the normal cells remain, the tumor cells disappear. I know this is only a movie, by the way, made by Andrea Wittig and Jean-Philippe Pignol long time ago, but it describes what we are doing. Nuclear medicine is also looking on the same way to irradiate cells with modern alpha emitters. But there's a big difference between boron neutron capture therapy and radionuclides. In Bnct, we can target with two independent, different mechanisms. One side, the drug who may go to the tumor, but may be a very high concentration to the kidneys. But as long as we irradiate only the head, we will not touch the kidneys, which is different if you use a radioactive source. You already have seen this night's slide from Paul. I like it very much because this was the initial issue that we tried to have combined clinical trial, MIT, Harvard one side and the EORTC on the other side, but finally money was not enough. This also has been shown, and this is another important aspect. This is an adenoid cystic carcinoma treated from our colleagues Hiratsuka and Aihara at the Kyoto University Research Reactor. And this is a tumor that does not respond to conventional radiotherapy. And here you see the follow-up after one single fraction. I have selected these three cases to explain the indications for Bnct and the number of patients that need Bnct. So in this early stage of clinical validation, we have to concentrate on situations that are not successfully treated by existing conventional methods. One of them are locally recurrent tumors after full regimen of conventional radiotherapy. We have already heard this example, from head and neck cancer. I see another nice indication for breast cancer, local recurrences on the chest wall after 2 times of radiotherapy, mastectomy. There are still these ugly cases of cancer en cuirasse. We have the melanoma of the mucosa where we can do nothing at all. We have metastasis from uveal melanoma, skin melanomas that are not responding on checkpoint inhibitors. And then the last tumor I just have shown is an example for radio and chemotherapy resistant tumors. In this case, an adenoid cystic carcinoma. There are also soft tissue sarcoma. And then there are still the tumors with very bad outcome, high-grade glioma, we just mentioned, but also malignant meningioma. If I add now all these patients or these indications I just mentioned, and I look on the Northern Hemisphere, which includes Europe, including Russia, China, Japan, and North America, then we end up with 1.3 million patients per year that are suffering from the indications I just mentioned, which is a huge, now I say the word, market for BNCT. Until recently, we were limited to the research reactors. That's, if I can say, my reactor at Patton, the Rolls-Royce of all the reactors worldwide. And I could use it 300 days a year, 24 hours a day, 10 years long. And I have treated in this 10 years 32 patients. And with such a performance, you cannot make clinical trials with including a statistical relevant number of patients. You can make proof of principle, you can make safety, but that's all. Now, the accelerators occurring, it's a shifting in paradigm. This is certainly not the most modern or actual image of the situation in Japan. There are actually 3 hospitals treating patients with BNCT, having a BNCT facility inside the hospital. And there are a lot of others under commissioning, under construction. If we look outside of Japan, then I can mention Helsinki. This is a nice facility in Helsinki. There are efforts in Russia, Pavia, 3 centers in China, Birmingham and Mayo Clinic and Knau at Pavia have signed contracts. So there is a huge activity. But BNCT will fail. Why? If we continue to work with standalone solutions oriented to make clinical research, all these centers will have different neutron qualities, which will result different biological effects. That was a problem we had also with fast neutrons. And we could not compare the results, and we could not copy what the others has done. So the results cannot be merged. You cannot make combined clinical trials. And actually, there is no coordinated effort to establish BNCT as a reimbursed treatment modality. And if you would like that BNCT one day is really used as a clinical modality in routine, it has to be reimbursed. Otherwise, if there is no money, you cannot continue. That how can we establish BNCT as a reimbursed treatment modality worldwide? The only way is we have to establish evidence. And evidence only can be established by carefully designed clinical trials, but also we need a result in a short time period. I mentioned here 2 years, maybe 3 years. But if we have, after 10 years, an answer on a question that was raised 10 years ago, the question is outdated and the answer may not interest anyone more. This means such kind of clinical trials cannot be done by a single institution. To overcome this problem, we have to create a critical mass from a group of BNCT facilities with the same equipment, following the same treatment protocol and working collaboratively. we need to train the staff in the same way. And this has to be done in an economically safe and sane way. We can do it by distributing tasks and sharing equipment and services. At this time, when we developed this in the frame of the German Society for Boron Neutron Capture Therapy, a pure without business interest, we said we have to change, and we created Bnct Global. The principle is there is a global service unit, and a lot of therapy centers are depending or controlled by this global unit. This allows us to make some kind of a blueprint policy, which means we are using the same hardware, the same drug, the buildings are the same, the software is the same. And this leads to what's called in the industry lean management and lean production, which means we can reduce cost and increase efficacy. Sorry, wrong direction. If now we have a Bnct treatment center treating patients, there are a lot of things they have to do locally. They have to treat the patients, they have to position the patients, they have to look for radiation protection, etc. But not everything, because the central unit can look for licensing, safety and quality assurance programs. Even treatment planning for Bnct could be done in a centralized way, the treatment protocols, of course, and so on. So by doing so, everybody who participates has benefits. First of all, costs can be reduced because we need less staff per center if we can delegate tasks. We can centrally procure the boron drug, which drops the price. And we can also centrally use cost-intensive computer capacities. There is comfort for the colleague who is participating, because regulatory authorities, it's not his task. There will be lawyers doing nothing else. We have uniform treatment protocols. So there's not the question how to treat this single patient. You have a protocol and you follow it. And very important, if we have a certain number of centers, there will be an expert for a problem in one of the centers. So these experts are at everyone's disposal. We are much more effective by merging data, which means rapid completion of clinical trials. And this leads to a reimbursement that becomes earlier possible. And by merging data, we also have the possibility to produce publication of higher impacts than if we try to do it alone with only 5 patients. There's also some specific benefits for the local partners, which is not only for the collectivity. And you have at your own a disruptive therapeutic modality. Nobody else has in the neighborhood. But you don't have financial risks because this will be covered by Bnct Global. Your staff will be trained. All this will increase appeal for patients. The therapy concepts are given. And at the end of the day, and it's very important if you are doing something which is really innovative, new, we have a shared liability. If you have 5 or 10 centers around the world doing the same thing, there are so many regulatory authorities looking on it that we are safe as an individual clinical scientist. This is a concept I would like to share with you, and I'm open for any discussion. And of course, if you go back to science, then please join the German Society, which already now has much more international partners than Germans, which is a pity. But so I go back and stop the interaction. I hope I was more or less in time. And I would like to switch directly to Professor Rock Mackie. Rock is really a rock in the modern radiotherapy. He is from Canada, And you all know if Canadians are doing some things, they normally do it in a better way than all the others. He was developing a very effective, very innovative modality to deliver conventional radiotherapy, the so-called tomotherapy, and he is still full of ideas. So I let him now the word. Please, Rob.
Thank you very much, Dr. Sauerwein. That was very flattering. I do have some disclosures. I am a founder of Leo Cancer Care, and I'll be showing some of their technology at this talk. I'm a board member of Cosylab, the sponsor of this meeting, and I'll show some of their technology. And I am a board member of Shine, which is making lutetium-177 Moly-99, or will make Moly-99, and has a neutron imaging facility in Madison, Wisconsin. So we've heard that essentially we're treating with high LET particles right inside the tumor cell. But what this needs is sufficient tumor to blood and normal tissue concentrations that will give a high enough dose to the tumor, but very low dose to normal tissues. A challenge is that all tumor cells must be labeled, and this can be difficult if the vasculature is compromised in the tumor. So it may be difficult to treat very large tumors. There may be some interesting technology to use a high energy gamma from the interaction as well for determining where and if the biodistribution is located. But really the key things I think are the tumor to background ratio and the amount of boron that you can get into the target volume. So, and if we look at all of the cancer journey of a particular patient, but also view it as an opportunity, then targeted agent development can be used in all of this area, from the diagnosis through conventional radiation oncology and radiopharmaceutical. And really the BNCT lies kind of interestingly between conventional radiation oncology and radiopharmacy. And the reason for that is the biodistribution issue is partly shared with radiopharmaceuticals. And while I agree that distant sites like the kidney and liver may not get significant dose, there is still an issue of what is the biodistribution that requires treatment planning. And of course, even radiopharmaceutical therapy is starting to be treatment planned. And so I think it's very important that BNCT also be treatment planned. So the requirements for a successful, if you like, a relaunch of BNCT is I think treatment planning. We will, if we want to be able to use this effectively, if you're buying an expensive piece of hardware, you want to be able to treat from multiple directions. You want to be able to treat where you want to treat, avoid what you want to treat. It should be a workhorse machine. It shouldn't be just confined to, say, head and neck and brain. There's likely to be other applications in the body. You do need in-room image guidance. You don't want the beam to miss the target volume. You want to have the neutron flux that you need. You would like to also be able to, if possible, to take into account that some parts of the tumor may be labeled better than other parts of the tumor. You need to then provide setup adjustment, which is important. And you, of course, need very reliable machine and patient quality assurance. All things that have been worked out in conventional radiation oncology and starting to be worked out in radiopharmaceutical therapy. So if you, I just wanted to point out, this is a system that has a vertical only beam line, which is actually very good for some sites, but it wouldn't be particularly good, for example, to provide lateral fields for the head and neck. Most accelerator-based systems, I think, will be using fixed horizontal beam lines just for the ability to site them in a conventional radiation therapy area. Here's a couple of examples of systems from two companies in the US. Sumitomo Heavy Industries can treat both upright and recumbent. So this, I think, is something that could be important for the field. Well, we have been developing at Leo Cancer Care a workhorse approach so that you can treat any site in the body in either the seated or perched position and have a CT scanner in the room to be able to determine if you're in fact set up correctly. There's an issue potentially in BNCT of the damage to the electronic components in a CT scanner, which are rich, of course, with electronics. But I think it's important to have CT in the site. This is showing results at the Paul Scherrer Institute for correcting the positioning using CT. And you can see that if you don't have CT, you're not going to be treating the site within a few millimeters, which I think is the goal of all radiation oncology. And they achieved this because they didn't have room on the gantry for a CT scanner. They actually had a CT scanner outside the room, which may be a solution for BNCT to avoid the neutrons as well, and actually transported the patient from outside the room to inside the room. It's also possible for hopefully this movie will run. Is the movie running? I'll assume it is unless someone tells me it's not.
Yes, it's running. No problem.
So basically, having a conventional cone beam approach with much less expensive hardware and more easily to replace if there's neutron damage could be a solution. Here we show a version of the upright positioning with a conventional 6 MV X-ray beam, but something like this could also be employed potentially for setup verification. So let me talk a little bit about the patient positioner. This is difficult because I wanted to show what the system looked like without the patient in place. But essentially, there are some non-motorized components. The backrest can be removed, armrest can be put in place, a vac-lock bag installed so that when the patient, and then of course the height of the seat, the angle of the seat, and the position of the heel stops put in place. And then the patient just simply sits down. So in terms of setup, it's very easy. Then the motorized shin rest moves into place, and immobilizing the knees and the shins really prevents the slouching that people worry about in upright radiotherapy. And then the patient is set up. The adjustment can be made with motors that are below the floor to provide elevation, left, right, front, back, yaw, pitch, and roll. So a 6-axis positioner to adjust the patient setup following imaging. And of course, imaging for a cone beam would be done rotating the patient. And I'm just going to show this movie. Of course, the treatment has to be done in the upright position by either having an upright positioner or having a fully recumbent positioner. This is a CT simulator that can CT sim both in the upright position as well as the recumbent position. And so this really allows then the CT sim to be used for conventional radiation therapy as well as for upright radiation therapy. This is a movie showing, a recent movie showing our prototype CT scanner moving down. Imagine it moving down over a patient that's upright, gets to the right position of the start of the scan, rotates, and then once the rotation speed is up, scans the patient in the vertical position. By the way, this is going to be a dual energy CT scanner. It is has the ability then to provide diagnostic quality images for CT simulation. It's also possible to use a synthetic MR from an axial CT scan to produce a pseudo MRI scan. And this can be used in a process to deformably register a patient's MR done in the normal recumbent position on a high field MRI, and then allow this to be used for radiation therapy treatment planning for an upright position where there isn't at this point a high field MRI. I should point out that there's two vendors in the world of upright MRI systems, and so it's perfectly possible to use those for treatment planning as well. Just like to point out that one of the interesting things of upright radiation therapy is it does allow a lot more space around the patient to put in other imaging systems. And Leo Cancer Care is cooperating with Cosylab to really kind of invent all the possible ways to provide image guidance, as well as flexibility in treatment modalities, and eventually, hopefully, BNCT as well. So in summary, you know, BNCT really shares, and this is myself rotating around. I'll just give you a picture of that as I talk. But it shares the biological similarities to radiopharmaceutical therapy, including an opportunity to have image-based planning. We really need to integrate into other radiation medicine modalities as well, and figure out how this modality can be used in conjunction, or even potentially replace others in time. It must be designed, I think, to treat the whole body to justify the expense of a system. The verification of the patient setup is essential, as it is in all forms of radiation therapy. Upright postures, either the seat or the perch, are now possible. And you can still use then conventional PET, CT, and MR systems to gather information on the target and normal tissue volumes. So thank you very much, and I will pass on the talk to the next speaker.
Thank you, Rob. I can.
Okay, very good.
Continue.
Thank you.
I only had problems with my micro. Sorry. Next speaker. Thank you for respecting time, and thank you for this really excellent talk, and especially thank you for having developed this technology. It's really great. I am now asking Hanna Koivunoro to continue. Hanna, I know her since quite a long time. When she started her PhD, she has done, in my opinion, she's one of the most experienced scientists in BNCT. She started with preclinical experiments. She has a huge experience in clinical medical physics for BNCT, and now she has switched to industry. So she is really someone who can teach us a lot. Hanna, please continue.
Thank you, Wolfgang, for a kind introduction. So I will speak about BNCT treatment planning today. And as Wolfgang explained, I was working for clinical trials in Finland for several years, and I performed most of the treatment plans for our patients. It was something like 250 patients we treated altogether in more than 300 sessions. So I'll share my screen now, and then I just need to start just a second. Okay, there we go. So first I will present parameters needed in BNCT treatment planning, and I will speak about BNCT dose. Then I will explain how BNCT treatment plan is actually done in practice, and including I speak about optimization. So in BNCT treatment planning, there are some special unique features. BNCT treatment planning requires modeling of nuclear interactions of neutrons within the patient. Typically, Monte Carlo simulations are applied for neutron and photon transport through patient model, which includes elemental composition of the tissues and also boron concentration of the tissues. Neutron dose calculation is a bit complicated too, because 4 dose components need to be taken into account. And in addition to that, there are organ and dose component specific RBE and CBE factors. There is no gantry at the moment for BNCT, so we need to work with fixed beam port. It complicates things a bit because patient needs to be rotated for optimal treatment orientation. So first about BNCT dose. As mentioned, there are 4 dose components in BNCT. Thermal neutrons that cause the therapeutically important boron dose, unfortunately, also interact with nitrogen and hydrogen in tissue, creating low energy intermediate proton dose, which we call typically either thermal neutron dose or nitrogen dose. And then thermal neutrons are captured by hydrogen in tissue, causing 2.2 MeV gamma dose in patients. In addition, in each neutron beam, there is also a little bit of fast neutrons that cause intermediate LET proton dose when neutron hits, kind of kicks off protons from hydrogen. This plot here shows physical absorbed dose components in function of depth in phantom. These are absorbed dose components, as I mentioned. Still, boron dose in normal brain is the highest dose component when calculated with typical uptake, boron BPA uptake of brain. Second highest boron dose component is photon dose, and then nitrogen and fast neutron dose components. Fast neutron dose component is typically very small. However, these physical dose components don't tell anything much about biological effect of BNCT before they are multiplied with RBE factors. We call RBE factor of boron compound or boron dose CBE factor, compound biological effectiveness factor, because it is dependent on the boron drug applied. So here are listed typically applied RBE and CBE factors. Sometimes some clinical groups use slightly different groups. Sorry about that, I have a dog here. And when we multiply its physical absorbed dose component with RBE and CBE factors, here on the right, you see biologically effective dose to brain tumor and normal brain. So the solid lines here are for tumor, and the dotted lines total dose and boron dose to normal brain. So when we speak about tumor dose, about 90% of the dose is resulted by boron dose, and only less than 10% from the other dose components. In normal tissue, the story is a bit different. So about half of the dose comes from boron and rest from the other dose components. So here is one example of head and neck cancer case. So unfortunately, mucosal membrane is very sensitive to boron neutron capture reaction when BPA is used as boron carrier. So here on very left, I have plotted the minimum tumor dose for this patient. Patient received around 20 grays to tumor. while mucosal membrane here received almost a little bit more than 12 grays, which is quite a high dose and really maximum tolerable dose for these patients. So tumor dose gradient in this case was about 2. So as mentioned earlier, boron concentration is monitored during the treatment based on blood boron concentration measurements. Here on the left is plotted blood boron concentration of a patient treated in Finland. BPA infusion started at time point 0, continued over 2 hours. After that, boron concentration starts to clear out from the boron. We take blood samples and analyze the blood samples at each of these points and start the first neutron irradiation about 90 minutes after end of infusion, and then second field a little bit after. At this time, blood boron concentration decreases quite slowly. On the right is plotted Professor Ono's method, which is applied in Japan and in Taiwan. So BPA infusion is again given in 2 hours, like pretty same dose as in Finland. And after 2 hours, BPA infusion continues during the neutron irradiation with half rate. So the system is quite different. And in both of these cases, the blood boron concentrations applied in boron in dose calculation are estimated based on literature. We use constant concentration ratios. So in tumor, typically we estimate that boron uptake is 3 to 4 times higher than in blood, and in mucosal membrane 1 to 2 times that of blood, and normally otherwise for normal tissues around same as blood. So then I speak about finally treatment planning itself. So in Bnct treatment planning, neutron source and the beam shaping assembly around the neutron source is typically modeled first with Monte Carlo simulation, and neutron energy and angular distribution are recorded inside the beam collimator or at the beam port. This is a cross-sectional view of the accelerator produced by Sumitomo Heavy Industry. So here is the neutron source. When proton beam hits the target that produces the neutrons, then there is beam shaping assembly that slows down the neutrons to epithermal region, and here is the beam port, and patient is placed here. Typically, circular beam ports are applied in BNCT from 8 to 10 centimeter diameter minimum up to 20 to 25 centimeters in diameter. Reference size in Finland is 14 centimeters and in Japan 12 centimeters. So in BNCT treatment planning, the patient needs to be modeled very first. So each organ are contoured for the model. The most important organs that need to be contoured are skin, bones, brain, lung, and air cavities, and brain including spinal cord. Typically, CT images are used for that task because it is possible to use all kinds of automated tools. MRI and PET images are often used for macroscopic tumor contouring, and at least in Finland, we used margin at least of 1.5 centimeters around the margin. Elemental composition of tissues is typically based on ICRU report 46, and homogeneous composition is applied for each region. These tissue elements need to include also boron 10, and boron 10 concentration should be modeled correctly within 20 to 30% for accurate neutron transport. As mentioned earlier, constant tissue to blood concentration ratios are taken from literature, and most often same boron concentration is assumed for normal tissues. only skin and mucosal membrane have more boron, and then of course cancer. And cancer boron concentration is sometimes taken from fluorine 18 BPa PET study before treatment planning. So because neutron field cannot be modulated, dose is very often inhomogeneous in large tumors in BNCT. For that reason, dose should be prescribed based on minimum dose to GTV. So here in this plot is a dose volume histogram for a patient who received BNCT one time in Finland. And actually, even though this GTV dose looks quite horrible if you compare to traditional radiotherapy, this patient had complete response after one time BNCT. So minimum dose to tumor was a little bit over 10 grays, but then there were parts in tumor where tumor received up to 35 grays in single fraction. All the time spinal cord dose was less than 4 grays. At the moment, we don't know optimal BNCT dose to cancer, meaning we don't know which dose is the minimum dose that would cure the cancer. And for that reason, dose is prescribed based on tolerance dose of normal tissues. For this reason, we give quite high doses to normal tissues in BNCT, and those treatment planning needs to be very accurately. And then I'll speak about plan optimization. BNCT treatment planning is actually quite simple. And at the moment, optimization is performed manually by rotating the patient in the beam using treatment planning software tools such as couch rotation, pitch and roll. In the optimization, sensitive organs such as spinal cord, structures of eye and mucosal membrane needs to be avoided. Now I mentioned these structures that are typical for brain and head and neck treatments because those are the most common targets of BNCT at the moment. In addition, we need to find shortest possible aperture to skin distance because a low energetic neutron field, but we are using for BNCT, lose their intensity very fast when we increase the distance. So it's something like 30% reduction in neutron beam intensity within 5 centimeters. Optimal plan is typically found with 1 to 3 fields in brain and head and neck cases. Opposite beams are needed for deep-seated tumors. And if the patient geometry changes due to position, treatment planning images should be taken in axial treatment orientation. So it makes treatment planning a bit complicated. So here I have listed some dose limits to normal tissues that we have used in prescribing BNCT treatment. So these doses are kind of in line with conventional radiotherapy if we speak about single fraction doses. And very often BNCT is given as ray radiation, so BNCT dose prescribed depends also on dose delivered from previous radiotherapy. So in mucosal membrane, we typically deliver maximum grays, sometimes a little bit more. Spinal cord only from 2 to 4 grays. And in case of newly diagnosed brain cancer, we have delivered something like 12 grays to maximum, dose maximum, and in irradiation of brain tumors, typically maximum grade 8 weighted grades. And then one unique feature for the treatment plan, dose plan needs to be scaled during the treatment based on actual measured blood boron concentration. So we don't actually know the monitor units that we need to deliver before we have verified the boron concentration on treatment day. And the planning is performed based on estimated blood boron concentration. So I'm showing here one plot which explains why it is difficult to know beforehand how much there is boron in the blood of that any specific patient. So here are several patients treated in Finland, or their blood boron concentrations plotted. And each of these patients received same dose of PPA, but the blood boron concentration levels during the infusion and after the infusion during the irradiation were very different. I'm sorry for this small, like this scale, you can't see the scale, but I think you can see the point based on this plot. So in the treatment plan, monitor units must be defined based on estimated blood boron concentration, and then during the treatment plan is normalized or scaled based on measured blood boron concentration. So then I'll tell something about the existing treatment planning systems. So in modern BNCT, obviously, treatment planning system must be approved for clinical use. And that's why during past recent years, several treatment planning software has been under development. Earlier, we used treatment planning softwares that were more, how would I say, academic systems, not approved for clinical use by any agency. So one also important thing is that because BNCT is often given after first-line radiotherapy, and sometimes BNCT is combined with standard radiotherapy, it is important that dose distributions from all modalities can be combined for dose planning, meaning that we can combine BNCT doses with, for example, photon therapy doses. So that's why it would be very good to have same treatment planning software for BNCT and conventional radiotherapy. So at the moment, there is one BNCT specific treatment planning software under development, which has been under development in Japan, Tsukuba University, for several years. It uses Monte Carlo code FITS, developed in Japan, as well as dose engine. In addition, research laboratories is in collaboration with Bnct accelerator vendors Sumitomo Heavy Industries, Neutron Therapeutics, and TAE Life Science. And each of these Bnct accelerator vendors have developed their own, or are developing their own dose calculation engine that can be used in race station. The new CURE dose engine by Sumitomo Heavy Industries has been already approved for clinical use in Japan. In addition to this, there is one more treatment planning software under development in China called New Boron Plan. And just as an example, also the features, BNCT features in race station. These have been in race station since version 8B, and the very next release will be 12A of race station. There are some more improvements for BNCT. So research has included in race station modality Neutron for BNCT treatment planning. There is a BNCT specific RPE model commissioning capability. Also NCT treatment machine commissioning, of course. BNCT treatment plans can be created, and couch can be tilted like we really need it for BNCT planning. And then of course for physical dose components can be computed, and RPE doses can be computed. And then my very last slide showing the treatment planning workflow planned for Finland, the clinical trials. So around 1 week before BNCT, we take pre-planning images, and we make like so-called pre-treatment plan based on these like normal, maybe diagnostic images. And that's done because we don't know the optimal rotation of the patient on the couch. So the very first images are taken in supine position. So after we found the beam angles, meaning patient angle, patient rotation on the beam, we take the axial CT images for final treatment planning. So patient is rotated to the position where the BNCT is delivered. I didn't mention that these are head and neck cancer patients first, and that's why the rotation is important. We calculate the dose plan again based on the axial rotation, and on treatment day, patient is of course positioned to the beam according to treatment plan, and then based on the blood boron concentration measured, MU monitor units are scaled the dose, and so that we deliver the dose that has been planned. So thank you very much for your attention.
Thank you, Hanna, for this excellent and really, every aspect covering talk. I'm always surprised how nice and how understandable you can show such kind of complex things. It's really great. I'm very glad that I can now introduce Akira Matsumura. Akira Matsumura is, from his formation, neurosurgeon, but he's probably the neurosurgeon with the most experience as radiation oncologist worldwide. He has made proton therapy, but especially also BNCT. But he always remained in the mainstream of medical application. He was a medical director of Tsukuba University Hospital for quite a long time, and now he is president of a university called Ibaraki Prefectural University of Health Sciences. I'm very grateful, Akira, said at this early morning in Japan, you are still talking to us. Please start your presentation.
Yes, thank you, Wolfgang, for your introduction. So I will share the screen. So do you see the slides?
Yes.
Okay. Yes, I will talk about the status of BNCT in Japan as requested from Cosylab. So in earlier times, we treated the patients in BNCT with the two reactors in Japan. One is Japan Atomic Energy Agency in Tokai, and the second one was in Kyoto University Research Reactor Institute. But these have been closed, shut down. The JR4 is shut down due to the earthquake, and the KURRI is now running, but will be shortly shut down due to the uranium consumption, I think. So I will introduce about the various accelerators developed in Japan. So as you see here, one is a cyclotron-based one, high proton energy, low proton current with a beryllium target, and this is a horizontal beam. I think this will be introduced by Sumitomo Heavy Industrial later in the second session. The second one is the LINAC, low proton energy with high proton current with a lithium target, and this is a vertical beam. This is put in the National Cancer Center in Tokyo. The third one is our system based on the LINAC with middle proton energy of 8 MeV and middle proton current from 2 milliampere to 5 milliampere, and barium target with a horizontal beam. So, and we have also electrostatic accelerator in Nagoya as well. So this is a summary of the Japanese accelerators. The first group is the based on the cyclotron made by Sumitomo Heavy Industrial, and I will talk about it later. It is already in the clinical application. And the second one is we have 2 accelerators in Japan made by CICS, and the National Cancer Hospital is now running the clinical study. I will talk it later. The third one is our University of Tsukuba lineup, and we are now started the preclinical study and now going to start the clinical study this year. And the fourth one is the Shonan Kamakura Hospital is now under construction and uses the system from Neutron Therapeutics. And the other 3 is now experimental or under development. So this is a Japanese map, and as you see here, the Southern Tohoku Hospital and Osaka Medical and Pharmacy University, OMPU, is running clinical treatment. And the University of Tsukuba National Cancer Center is preparing to do the clinical trials. And other is Edogawa Hospital, Shonan Kamakura Hospital. You will hear about these, I think, later. So this is our University of Tsukuba Proton Center system. This is IBNCT project, and this is accelerator and irradiation room, treatment room, put in the building of Ibaraki Neutron Medical Research Center at Tokai. So this is a proof of concept accelerator. In the future, we will put this machine into our Proton Medical Center if we have approval from the PMDA. So this is the Shonan Kamakura General Hospital, and this is their new building. And they put proton therapy and also BMCT from Neutron Therapeutics in this building. This is a very unique new concept from Fukushima SIC Applied Engineering, and they have 10 small neutron source made by silicon carbide, and they will put this into this Kyoto Prefecture University of Medicine Rome BMCT Center. But I don't know the details how this work. But this is a very unique system in Japan. This is a neutron beam profile, and this is a JR4 profile, and this is a KUR. And as you see here, the accelerator from Siemens is here, the black one, and the dynamitron in Nagoya is the green one, and the IBNCT in Tsukuba is the red one. And the beam profiles is slightly different. And as you see here, this is a 10 keV. And as compared to the reactor, accelerator have slightly higher neutron energy compared to the reactors. And the Siemens has a slightly higher neutron energy compared to other accelerators. So BNCT as a insurance covered treatment. This is a news release from Stella Pharma, which produces the boron compound called steboronin. And this is a news release from Southern Tohoku Hospital, where they did the world first treatment covered by the insurance in recurrent head and neck cancers. This is a summary of the clinical trial. So, sorry. So first, the cyclotron-based BNCT accelerator by Sumitomo Heavy Industrial combined with the boronated compound steboronin by Stella Pharma. And they started the clinical trial in 2016 for recurrent head and neck cancer. And they got the approval from PMD, this is Japanese FDA, in 2019. And then they got the approval from Central Medical Council of Ministry of Health, Labor and Welfare in 2000 February. So when it goes through the Japanese FDA, they go to the Central Medical Council, and then it will be covered by the National Health Insurance. So in 2020, June, this was approved by the Japanese government that will be covered by the National Health Insurance. And the medical cost is here. So irradiation fee for the accelerator is 2.4 million Japanese yen. And the boron drug, one package is 440,000 Japanese yen. And we usually use 4-pack for adult. So it's about 1.7 million Japanese yen. In total, it's about 4.1 million Japanese yen. And in Japanese health insurance, the patient has to pay 30% of the total cost. But afterwards, there will be some refunding depending on the annual income of the individual patients. So the patient doesn't have to pay all these 30%. So some will be covered by the government. And besides the recurrent head and neck cancers, there are several ongoing trials in Japan. The first one is a recurrent malignant glioma. This is closed in 2020, and this was a phase 2 single arm headed by Osaka Medical and Pharmacy University. And they got the median survival time of 18.9 months in this recurrent malignant glioma. This is a very good result, but the PMDA is now requiring comparison with the historical data. So now they are comparing this MST with the historical data. And we hope it will be soon approved by the PMDA, and that we can treat the recurrent malignant glioma with the health insurance covered treatment cost. The other one is the malignant melanoma and angiosarcoma. This is a phase 1 ongoing trial headed by National Cancer Center. And the third one is the malignant meningioma. This is also running from 2019. This is a randomized controlled trial of 18 cases. And this is headed by Osaka Medical and Pharmaceutical University. So we hope that these type of tumors will be approved, and the cost will be covered by the insurance in the near future. And finally, the approval for Bnct accreditation. So the Japanese Society of Neutron Capsule Therapy launched a certificate system for board certification for Bnct treatment doctors. So there will be written tests in treatment procedure, related technical issues, therapeutic effect, side effect, and pre and post Bnct management. So this test covers 4 areas. This is physics, engineering, biology, chemistry, and also each clinical specialties. So prerequisite for eligibility requirements for candidate for examination is that the candidate should have the Japanese medical license or a surgeon with dental license. So there should be no penal provisions of medical or dental practice, and the candidate must be JSNCT member at the time of application. And he or she has to have a clinical experience at least 3 years in their specialty. And to go to the examination, they have to have clinical experience required for accreditation of the BNCT as BNTCT physicians. So that means they have to experience 5 cases of BNCT treatment facility with no knowledge of clinical indications and also decision of the treatment planning, participation in drug administration and neutron irradiation. So when they have 5 cases experience, they can get into the written test. And after having this board certification, there will be renewal of the accreditation. So he or she has to have 20 cases experience within 5 years, and also had to attend at least scientific meetings of JNCT or ICNCT in the past 5 years. So from the homepage, currently 30 physicians has been approved. There are 15 neurosurgeons, 9 radiation oncologists, 3 oral surgeons, and general surgeon, ENT surgeon, orthopedic surgeon, each 1 person. I think every year we have the test, and I think this number will increase dramatically in the near future. So as summary, in Japan, several types of accelerator-based BNCT and equipment and the BPA-based drug has been developed. And in the combination of accelerator and boron drug, clinical trial for recurrent head and neck cancers has been successfully performed and has become a national health insurance-based general treatment for recurrent head and neck cancer. So I have presented, there's other clinical trials for malignant melanoma, angiosarcoma, recurrent malignant glioma, and malignant glioma. These are in progress. And as Wolfgang also stressed, for expanding clinical indication, we have to consider about the global clinical trials, even if it's some difference in neutron beam or drug administration, but we should consider about this seriously. So thank you for your attention.
Thank you, Akira, for this really very promising presentation of what's going on in Japan. You're really in front of all of us, and I'm admiring how our colleagues in Japan were able to make such kind of important work in such a short-term period. If I look on time, we are a little bit out of time, and I would suggest that we are not making an oral discussion. Vanya, if you agree, because we have foreseen 3 hours, which is a very long time period, and in Japan it's now 1 o'clock in the morning. So I suggest we continue directly with the second part on industrial aspect of BNCT, moderated by Nick Schroeder. But prior to close my first session, I would like to thank everybody, all of the speakers, but especially Cosylab for having arranged this really interesting and important talk. Thank you.
Thank you, Wolfgang, for the introduction, and thank you again, everybody, for participating in this extended session. I do want to say that please use the Q&A panel. I keep on monitoring the Q&A panel. It's actually pretty good, and we will make sure that these questions get answered and circulated also via email. So this part of the session, when we put the seminar together, we realized there's about 12 different groups involved in manufacturing or providing various forms of BNCT equipment, but we invited 3 groups, Sumitomo, Neutron Therapeutics, and the TAE, to talk to us a little bit about the technical aspects, the technical and industrial aspects of BNCT. So again, I'm not going to waste much time. It's going to be recorded. People can watch it again. And so let's start off this industrial aspects of BNCT session, and we ask Toshinori Michimoto from Sumitomo to give us an overview of their technology.
Thank you, Chairman. So I'd like to start my presentation. So thank you. I'd like to introduce nuclear BNCT system. This is a Sumitomo BNCT system. Yes, so at first, I'd like to introduce the Sumitomo heavy industry a little bit. So its vision and the mission in the healthcare segment. So vision is create a society where people can live live long and healthy life, even if they should from cancer. And its mission, they contribute to the diagnosis and the treatment of cancer with medical accelerators. And in the area of the cancer treatment, we provide proton therapy system and the neutron boron capture therapy system according to the stage of the disease. So I will show you the experience of Bnct system. So Sumitomo constructed 3 systems. Now 2 systems are for clinical use. 1 system is for non-clinical use. 2020, so the system get the medical device approval. Since that, so more than 160 patients treated. And this is a system where it installed. So first the system is installed in the Kyoto University. Yes, 2009. And second system is installed in Southern Tohoku General Hospital in 2014. And the third system is installed 2018 at the Osaka Medical Pharmaceutical University. And Kyoto University system is now used for only non-clinical use. And the Southern Tohoku system has 2 treatment rooms. So now we have 3 treatment rooms for the clinical use. This graph shows the this slide shows the number of the treatments per year, including clinical trials. And in this period, so we go the clinical trials, and after that, so number is improved, increased after the medical device approval and the insurance start. And for the BNCT, and so we start the development of the system about 2007, and from 2012 we started the clinical trials with the Japanese accelerator and the drugs. And the drug is supplied from the Stella Pharma company. And in 2020, so we get a medical device approval and also insurance covered in Japan. So I'd like to skip this slide so everyone knows very well, I think. And so I will introduce to the nuclear BNCT system. This shows the nuclear BNCT system. This is an example of the layout. So So there's a cyclotron, and the beam lines and the beryllium target is here, and the treatment room is here. And next to the treatment room, we have a preparation room, and patient is set up in this room and moved to this room, and then irradiated and treated. After that, then go back to the preparation rooms. So this is the photo of the treatment rooms. So I'd like to show you some video. So it shows the treatment flows. So first we start the preparation. So preliminary treatment plan is prepared. The patient setup is determined based on the preliminary so you see, so shell is prepared. And this is sorry. And finally, the final treatment planning is done. Infusion brought drugs is start 2 hours before irradiation. So we can use a bed or chair instead of the cancer. The patient is automatically transported to the treatment room. So we measure the drug boron concentration before the irradiation. And then treatment started. Treatment is about from 30 to 60 minutes. So this is a treatment flow. And so I will show you the feature of the nuclear system, Bnct systems. So there are 5 features: one cyclotron, target, and treatment table, and the patient transport system, and treatment planning system. Cyclotron, for the cyclotron, many cyclotrons are used as medical accelerators to produce a radioisotope for PET on the proton therapy. More than 200 cyclotrons are installed in hospitals. For Bnct use, high-power cyclotron was developed. It can accelerate protons to 30 MeV with current of 1 milliamps. And the beryllium target produces a neutron with high stability and safety. And in 30 MeV proton beam, so beryllium target is good for production of the neutrons. Patented neutron dose control by proton current monitors for the accurate dose deliveries. More than 1 times 10 to 9th per square cent per sec thermal neutron flux will be get. And the patient transport system, and the patient setup is done in the preparation rooms. The system transports the patient to the treatment room. After treatment, the system transports the patient safely back to the preparation room, reducing the exposure of medical personnel. And so we have 2 tables. So one is seated and the other is supine. So this is applicable to various regions. The seated is for head and neck cancer, and the supine for the brain. So this can minimize the air gap to make the most neutron flux. And for the treatment planning system, so we developed a FITS-based dose engine by Sumitomo, intuitive GUI by research stations, validated in combination with the nuclear system. And the nuclear dose engine approved in Japan as medical device March 2020. And also recently, new extended collimators approved. Yes, and the extension is 5 centimeter and or 10 centimeters. That is for easy patient posture while the keeping neutron flux. And for the clinical trials, so head and neck cancer, so unresectable, recurrent, or locally advanced. They already approved, and we use the drug from Stella Pharma, also approved. Well, malignant glioma, or type of the cancer is recurrent, and phase 2 clinical trial completed. Now discussing with PMDA, so Japanese authority. For the malignant meningioma, so recurrent cancer, and phase 2 recruit completed. And head and neck cancer, phase 2 clinical trial. So result is shown, Dr. Hirose, the radiotherapy and oncology, and the Sumitomo system and Stella PPA is used. The response rate is 71%, and no grade 4 or 5 serious adverse event except for hyperamyloidemia. Thank you for your attention.
Great, thank you very much. Excellent presentation and cool animation. So let's move forward to our next industrial aspect presentation by Dr. Liz Rezek from Neutron Therapeutics. Liz, please continue.
Great, thank you very much. Let me share my screen. All right, you can see my presentation. Excellent. So first, I'd like to thank our Cosylab colleagues for the invitation to speak today. I'm very excited to be here and really thrilled to see how much interest there is in the field of BNCT. Although I have a background in cancer biology, I myself am relatively new to the field of BNCT, having just joined Neutron Therapeutics this past August. But I have to say that the energy in the field is palpable, and the momentum is definitely growing. So that's really exciting. It's an exciting time to be in the space, and I'm honored to join those of you who've been pushing this field forward for decades. For those of you who aren't familiar with Neutron Therapeutics, Neutron was founded in 2015 in Danvers, Massachusetts, to bring proton accelerator technology that was originally developed for industrial applications to bear on the problem of clinical BNCT. Our team is comprised of engineers with decades of experience in pioneering accelerator technology, along with a medical physics and clinical team with more than 20 years of experience in driving BNCT research forward in Helsinki, Finland. I was asked by our Cosylab colleagues to speak today on the industrial considerations of BNCT from NT's perspective, and I thought I would focus on some of the aspects of NT's solution that address key challenges for the operation of a BNCT center in the clinical setting. There's nothing unique about these challenges. They're challenges that must be solved by any accelerator manufacturer developing a system for BNCT. However, as an outsider coming into this space, I have to say that I've been extremely impressed by the way that the Neutron team under Ted Smick's leadership has thought through each of these challenges and come up with elegant practical solutions that together make the NuBeam system a complete solution for hospital-based BNCT. NT's NuBeam system is the world's first and only BNCT system that conforms to all IAEA established international standards for clinical use of neutrons, and our mission is to provide powerful new cancer treatment solutions to patients with no other options. As we've already heard earlier this morning, BNCT began in the setting of nuclear research reactors, where groundbreaking research was done to show the power of combining neutron irradiation with targeted boron drugs to specifically target tumors at the cellular level. However, it's easy to appreciate why the clinical utility of this approach was limited and why progress in the field slowed significantly as research reactors ceased operations. Today, I'd like to tell you how NT has approached the problem and why our solution is uniquely suited to bringing BNCT into the mainstream and to making it a reality for patients with no other options. The NT team had the benefit of starting with a mature accelerator technology with a track record of safe and reliable operation in the industrial setting. Their challenge, therefore, was how to address the unique needs of the clinical setting, including incorporating imaging and patient positioning solutions, dose calculation and treatment control software to provide a complete treatment solution. The entire NuBeam system was designed to fit within a similar footprint to a traditional radiation therapy suite on a single level, which we believe is key to widespread adoption in existing hospitals where square footage is often at a premium. Special consideration was given to radiation safety and exposure limits for staff who operate the system day in and day out. Image-guided patient positioning was designed to streamline the workflow and allow more patients to be treated in a typical workday. In addition, a great deal of thought went into the service and maintenance aspects, a necessary part of any clinical system, to ensure that it can be done with minimal downtime and risk to staff. Finally, the industry-leading power of the NuBeam neutron source allows for significantly shorter treatment times for patients and as few as 1 to 2 treatment fractions. As I mentioned, the NuBeam suite is a full BNCT solution designed for clinical use, incorporating all of the components necessary for imaging, treatment planning, patient positioning, and treatment in a safe and streamlined work environment designed for high throughput workflows. NuBeam is comprised of a 2.6 megavolt, 20 milliamp proton accelerator, a beam line, beam shaping assembly, and a neutron generating target that also provides shielding to protect the patient and staff. A positioning robot and in-room CT scanner allows for image-guided treatment in real time. In the rendering here, you can see what the typical layout of a suite would look like, with high-density concrete shielding surrounding the beam line and treatment suite for safety. Imaging, positioning, and treatment are fully automated, and the patient and clinical staff are fully protected both during and after the treatment. Again, note that the entire system is housed on a single level, which makes incorporation of the NuBeam suite into existing hospital facilities more straightforward than other approaches. To give you an idea of what a typical suite looks like, these are images from the NuBeam suite at Helsinki University Hospital, home to Europe's first accelerator-based BNCT installation. The system is currently operating on a daily basis for verification and validation testing, and the first clinical trials are now expected to begin in late 2023. Those of you who have been following the field of BNCT will know that we've been working towards this key milestone for quite some time. And as with any first installation of a new technology, there have been some unanticipated challenges that we've had to work through. However, I'm happy to report that these have been resolved and that V&V testing will be completed later this year. Helsinki University Hospital has provided us with invaluable input and feedback into the design characteristics of a clinical BNCT system, and we are extremely grateful for their close collaboration and support. Installation of Neutron's second new beam system is currently ongoing at Shonan Kamakura General Hospital, a part of the Tokashukai Hospital Group in Kanagawa Prefecture in Japan. The new beam suite is located in Shonan Kamakura's new Advanced Medical Center, adjacent to their proton therapy suite, and the facility itself is a gorgeous, state-of-the-art hospital, the flagship hospital of Tokashukai Group. Travel restrictions to Japan have certainly made our job more difficult during the pandemic, but we do have boots on the ground there, and we'll be sending additional team members to complete the installation as soon as the borders reopen to travel in March. Installation is expected to be completed in the fall. As I mentioned, facility footprint was a key consideration for Neutron in developing the new beam suite. This is an important factor in ensuring that the suite can be accommodated near supporting treatment facilities in the main hospital building to reduce costs and streamline workflows. However, the new beam suite can also be designed as a standalone facility with ancillary rooms placed adjacent to the treatment suite depending on the needs of the hospital. A typical new beam facility is placed on a single level with no space or access required below the bunker floor or above the suite for service or operation. The facility requires a minimum height of just over 4 meters and has a footprint of 2,500 square feet or 230 square meters for the suite itself, with an additional 1,200 square feet or 110 square meters of ancillary space. Should supporting facilities be required for a standalone installation, these would be designed in conjunction with the hospital to suit their specific requirements and space constraints and can be located adjacent to, above, or below the new beam facility itself. The safety of the staff operating and maintaining the facility was of critical importance in the design of the new beam suite to minimize disruption to workflows and maximize throughput. Our design target for staff exposure was 20 microsieverts per hour during normal operation. As shown here in the simulation, dose rates in the treatment room drop below this limit by 1 minute after beam stop, allowing the staff to enter the room almost immediately after treatment is complete, with no special procedures required. We've confirmed these projections in the Helsinki new beam treatment room using actual activation measurements following neutron beam testing runs. Safety during target exchange was another key consideration during the design process. New beam's proprietary target design offers an exceptionally long lifetime for our lithium targets, allowing close to a year of operation at maximum utilization before targets need to be exchanged. Spent targets do need to be handled with caution and stored for a period of time before they can be reconditioned and reused. To make target exchange as safe and routine as possible, new beam utilizes the patient positioning robot to remove the spent targets, place them in the lead-lined storage bins for disposal, and fit new targets. This elegant solution eliminates technician exposure, reduces impact on treatment schedule, and reduces the cost of target exchange. The picture shown on the right is a target exchange demo unit located in our Danvers, Massachusetts facility, and I welcome any of you who are interested to come stop by and see it. Treatment workflow is a key component of the new beam system, allowing for potential throughput of more than 2,000 treatments per year at peak utilization. To achieve this, new beam utilizes a 7-axis ceiling-mounted robotic positioning system for both imaging and patient positioning to ensure fast and accurate treatments. The images in the upper right-hand corner show the patient in the treatment position and imaging position respectively. The image on the bottom left shows a rendering of the treatment suite in Helsinki, and the image on the bottom right shows the actual installed solution. Automated imaging and patient positioning allows for more accurate treatments without the need for tedious manual measurement and positioning solutions. One of the most important considerations for the team in designing new beam was minimizing the cost of patient treatment by maximizing the power and quality of the neutron beam for the shortest possible treatment times. New beam was designed to operate between 30 to 40 milliamps, far higher than other competitive systems. This enables us to utilize more stringent beam quality metrics for improved patient safety while minimizing treatment times. Keeping treatment times to 20 to 30 minutes greatly reduces patient discomfort and immobilization issues while increasing throughput and allowing for greater optimization of boron carrier kinetics. Shorter treatment times and fewer fractions makes the treatment process less onerous for patients and makes medical tourism-based BNCT treatment a possibility for patients not located near a cutting-edge cancer treatment facility, increasing potential profitability for the hospital. NuBeam is the only BNCT system that meets IAEA beam quality recommendations. Unlike other competitive systems, our industry-leading neutron flux allows us to filter out biologically harmful fast neutrons greater than 10 keV to minimize damage to normal tissues while effectively targeting tumor tissues with high flux therapeutic neutron irradiation. This capability puts NuBeam in a class of its own among BNCT treatment systems. NuBeam has a track record of reliable high-duty operation at our Helsinki University site, where we've demonstrated on-target proton current for extended periods of time up to 20 to 40 hours per week for 16 consecutive months, and as high as 80 hours in a single week. We are continuing to operate the Helsinki machine on a daily basis as we carry out our V&V testing in support of the regulatory approval process. No other system has demonstrated such robust and reliable operation, making NuBeam the obvious choice for high throughput BNCT clinics. I hope I've managed to give you an appreciation for the key features and design considerations that went into making the NuBeam system the world's only safe, high productivity BNCT system for the hospital setting. I'm extremely excited to be a part of the team that will bring this product to market. The Neutron team is passionate in our mission of bringing hope to cancer patients worldwide who currently have no other options. Finally, I would like to offer our sincere thanks to our key partners who have been instrumental in our success thus far. In particular, thank you to Cosylab for their support and collaboration and for organizing this fantastic event. Thank you very much for your attention, and I'm happy to answer any questions in the Q&A chat box.
Great, thank you, Elizabeth. I just want to also again encourage people to participate in the Q&A, and also for the people that the questions are addressed to kind of address those questions as we go along. There will be time after as well. So we talked technology now, accelerators, everything. Let's hear a little bit about pharmaceuticals. And so we now have Seppa Pakala to talk a little bit about the BNCT drug development. He's also from Neutron Therapeutics. And then after that, we'll hear from TAE Life Sciences, Rob Hill. So Seppa, let's hear from you.
Hello, everyone. I hope you can see my slides.
All good, yeah, we can see it.
Yeah, okay. In the agenda, it is stated that I represent Neutron Therapeutics, but I have to say that I'm also the chair of the board of directors in 10 Boron. Many thanks to Cosylab, who gave me the opportunity to talk in this very nice webinar. Let me first remind you about some of the requirements of boron carriers. Boron carriers differ from other drugs because they are not expected to possess therapeutic effects, only to be able to carry boron to target cells and to be non-toxic. So they need to be specific to the cancer cells in the target tumors. Boron in tumor macrophages or other supportive cells will not yield therapeutic effect. Treatment will be effective only if, with the carrier, we can achieve high boron concentration in target cells, and the boron retention time is long enough to allow efficient neutron irradiation. The boron load to normal cells should be very low, and clearance from blood should be rapid to avoid harmful effects in normal tissue. Clinical trials with current boron carriers have shown that therapeutic effect can be achieved if boron content in tumor is at least 3 times higher than in normal tissue. If we can achieve much higher tumor to normal tissue ratios, we could shorten the irradiation time, and then we would be able to treat more patients per day. There are some requirements for clinical use which have not yet fully been met with current carriers. To be widely used, the drug must be produced according to good manufacturing standards before it can be registered. Manufacturing in GMP facilities is expensive and out of reach for academic groups. Currently used BPA fructose is manufactured in hospital pharmacy separately for each patient, which is time consuming, and quality of each batch must be guaranteed separately. For brain tumors, the ability to pass blood-brain barrier would be beneficial. Opening of blood-brain barrier with mannitol or other means may result in uneven distribution of the carrier and in poor treatment result. The verification of the boron content in the tumor during irradiation is recently not possible, but would be a real asset for optimal dosing. We can only measure 18 fluoride labeled BPA before irradiation using PET scanning. This can verify the uptake of the carrier and give some indication about the boron concentration in the tumor. However, this does not verify the dose in cancer cells during irradiation because the dosing for PET and therapeutic use are different. The result may be that boron concentration during actual irradiation is not what was anticipated by PET result. So there is an area need for studies optimizing the dosing of BPA. But even before we are in clinical phase, we need to overcome some regulatory hurdles. Different economic areas have different rules for combination of a medical device and a drug for the therapeutic use. In Europe, the device and the drug will be assessed separately, but in Japan and in USA, they will be assessed together. This means that once you have a registered boron carrier in EU, you can use it with any approved accelerator. In Japan and USA, you have to go through a regulatory process, which is combination of a boron carrier and an accelerator. Also, the need for clinical studies may vary. For example, the extent of preclinical studies needed before biodistribution studies. Also, the IRBs in different countries may consider differently the ethical aspects of the studies on non-therapeutic drugs in volunteers and patients. Boron carriers have been developed in research laboratories for a long time. Numerous compounds have been studied, but only 2 have passed on for clinical studies, and only 1 is registered, BBA sorbitol in Japan. The reason for this lack of molecules for clinical use is due to the fact that I call authors declare no conflict of interest phenomenon. Since there has been only few centers, there has not been any commercial interest in boron carriers. Consequently, there has not been enough funding and resources for advanced drug development and GMP manufacturing. But I believe this is about to change since the number of installed accelerators is rapidly increasing, and several BNC centers can produce clinical results more efficiently. Increasing the indications and number of potential patient populations will make BNC commercially more viable. This should make pharma companies interested in boron carrier development. However, today I'm aware of only 2 companies which are actively developing boron carriers: TAE Life Sciences and 10 Boron. Here I can only comment on publicly available data, but since the next speaker will give you an update on TAE's progress, I will skip this slide. I will briefly describe 10 Boron's pipeline. The company has 3 different proprietary molecules in different development stages. An antibody boron conjugate for HER2 positive tumors, which is about to enter clinical phase this year. A BPA-like non-specific carrier, which is in preclinical development phase. And a glioblastoma protein targeted carrier in early development phase. This is a schematic picture of 10 Boron's TB0010 molecule. The antibody is linked to a carbohydrate boron complex, which has high boron content, over 1,000 boron atoms per molecule. Thus, it has been able to produce high tumor to blood ratios in head and neck tumors in xenografted mice. Because of the high boron content in tumors, the irradiation time can be short, and long retention time in tumor makes scheduling of irradiation easy. This molecule is about to enter phase 1 studies later this year, as I mentioned. Although current boron carriers may not be the best solutions for a long run, they have been able to demonstrate the proof of concept of Bnct and provide very promising results in several indications. There are already business opportunities with BPA-based Bnct, which may produce revenues while new carriers are emerging from clinical studies. If one wants to be the leader on this field, one must start now. Thank you for your attention.
Great. Thanks, Seppo. Thanks for being very short in time. We are making up good time. So as Seppo mentioned, our next speaker would be Rob Hill from TAE Life Sciences. And so development status of the TAE Bnct system. So Rob, let's hear from you.
Thank you so much. Can you see my screen?
Yes, looks good.
Okay. Well, I want to thank Cosylab for the opportunity to present in this meeting. And I want to thank the other speakers for being on time. As I'm the last presenter, I really appreciate having the full time slot. So my name is Rob Hill. I'm the COO of TAE Life Sciences. I'm going to give a little bit of an introduction to our company, excuse me, our products, and also an update for people who are maybe more familiar with us so far. So our company is really focused on Bnct. That's our principal focus, our area of interest. And we're intrigued about it because we consider it to be an elegant combination of biological solutions and physics-based solutions. So how we got started as a company is we're a spinoff of the world's largest privately funded fusion energy program called TAE Technologies. They are focused on solving the world's energy problem, limitless clean energy for the world. They had developed a neutron accelerator as part of their program, and we spun out our company in May of 2017 to commercialize that neutron accelerator for Bnct applications. Early on, it became very apparent that the accelerator was only a piece of the solution, and that, as many of the presenters on this panel have said previously, really new drug development is needed to unlock the full potential of Bnct. So we opened our pharmaceutical lab and hired many veterans from the pharmaceutical industry in late 2017. And so our focus is to develop the full Bnct solution for our customers. We're a very young company. We're humble, but at the same time, we have a lot of great industry veterans, both from radiation oncology, which is my background. I've been in the field for 30 years, as well as pharmaceutical development. We've also got great support from investors. We have had $95 million in funding so far to date. So let me first start talking about the biology aspect of our company. So we are focused, as the last speaker described, we're focused kind of in two primary areas. One is developing small molecules that are in many ways similar to the current drug BPA. And we're also working on newer generation technologies we call antibody boron conjugates, ABCs for short. With regard to our small molecule program, we've now developed several product candidates I'm going to talk to you about, and we've done animal tests for these candidates. So these are really, we're moving these toward the IND stage, really looking to start to get these in clinics and start to use them on patients as soon as we can. For antibody boron conjugates, our focus there has been on the linker technology, delivering 100 boron atoms per antibody. Though that's an earlier stage activity, we're still discovering the right approaches to use there. So quite a bit of patents in that field, but still a little bit further from using it in the clinic. I'm going to spend most of my time today talking about the small molecule program. So we made excellent progress on this. We have several different amino acid analogs that we've developed. We have 4 product candidates, all of which use a similar transporter mechanism that BPA uses. Similar to what the other speakers have said, BPA is an excellent drug. It's a good starting point, but it has some key limitations. And so what we do in evaluating these new drugs is we compare how they perform against how BPA performs. So we've done extensive testing in our lab with skid mice to determine how well these drugs will perform. One of our product candidates has longer retention in tumors compared to BPA. That's a nice feature. And I'm going to share with you some data that we've acquired on these candidates. So 2 of our candidates, TC220 and TC221, we've tested in a range of in vivo models to just determine the robustness of the drug, of the approach. We've done it in FADU, in MeWo, in A549, Detroit 562, HCC, and F98, a rat glioma model. And we see excellent and significantly more boron uptake in these models compared with BPA. And of course, there's variability between the different models that are used. Those that have the highest LAT1 expression tend to have the most boron uptake. And one of the things that we surmise is that LAT1 expression is going to be important in determining how well patients respond to BNCT, both with BPA and with these new drugs. It's a big area of research for us, kind of understanding variable LAT1 expression. The key takeaway from these exercises is quite encouraging. Our new compounds consistently were able to deliver about 2 to 3 times more boron than BPA has done in all the models that we've tested. So I think this is potentially significant in terms of certainly dose escalation is possible if you can get more boron into the tumor cells. Also, shorter treatment times are practical, treating tumors at deeper depths. So I think there's lots of advantages to delivering more boron to tumor cells. Reinhard asked in the questions earlier in the meeting, I know he mentioned he had to leave. He asked about how do we arrive at more specificity in boron delivery? So how do we get to a point to where we are delivering boron more specifically to the tumor? So we've studied this as well, just highlighting in this case a single tumor model, FADU. On the left-hand side of the slide, we show the tumor to blood ratio for 2 of our product candidates. We also experiment with different dosing levels for all of them and comparing against the performance of BPA. And we also looked at tumor to brain ratio. The key takeaway here is that with TC221, one of our candidates, we're able to achieve a significantly improved selectivity of delivery of the boron. So when it comes to the blood, tumor to blood ratio, we're able to achieve an 18-to-1 ratio for TC221 versus a 7-to-1 ratio in BPA. So you may be wondering about that. Previous talks today talked about maybe a 3 or 3.5 ratio between tumor versus normal tissue. Those are in humans. This is all in a skid mouse. So the behavior is a little bit different, but the key takeaway for us is the differential performance compared to BPA. So with the blood ratio, we're able to achieve a 2.5x improvement over BPA, and with respect to the brain, a 3.6x improvement. So we're very encouraged about this. We think this has the potential to really reduce normal tissue doses in BNCT. So the work that I mentioned before is for 2 of our product candidates, and it's mostly based upon work that we've done in our lab in Santa Monica here in California. We took 2 of the compounds though, and we actually used them in our collaboration at Kyoto University, where we did a Bnct experiment. So what we did was we took their established protocol where they take mouse colon cancer allograft CT26, and they would treat them for 12 minutes for standard, the standard treatment that they normally do with BPA. We ran an experiment where we tried to treat these cases with half the treatment time to see whether these drugs that what we saw in our lab, that they were getting much larger amounts of boron delivered, whether that bore out, the results would bear out in a BNCT experiment. So there's kind of 3 curves here where you see the tumor is growing significantly over time. That's the control group in red, where there's no drug given, no irradiation given. There is the green curve, which is the group that received neutron irradiation, and then the gray curve, which received BPA. And then our new compounds were the orange and blue curves. Those curves really inhibited the tumor growth going forward. So it was a successful experiment to show that the drugs would be effective in preventing the tumor growth. And in fact, on day 40, there was no evidence of tumor left in the mouse, only scar tissue. So this was a very surprising outcome that our partners in Japan did not expect to see from this, but we were highly encouraged by the results. So that gives you a sense of some of the new drugs that we have in development. And we look forward to continuing to test these drugs and sharing the data going forward. Switching gears a little bit to talk about the physics aspect of the system. So we're installing the first accelerator now with our partnership with New Boron Therapy Systems. They're active in the China market. We've now installed the system in Xiamen. Just to summarize a little bit, the system, the picture on the top right shows the BNCT center that's been constructed. In the middle of the right-hand side, you can see kind of a schematic, and the bottom shows the accelerator. The facility is designed as a 3 treatment room facility. Our accelerator kind of characteristics is we have a tandem accelerator. The beam starts out as a negative hydrogen ion beam. We then pass it through the accelerator where we scrape the electrons off the hydrogen atom, which makes it a proton beam, and we accelerate it to the megavoltage energy range. Then we use magnets to steer and focus the beam to the appropriate treatment room. A key facet of our design is the lithium targets are easy to replace. They're at the end of the beam line, and they're used to generate the neutrons. In terms of some early results from this installation, so we just installed it in 2001. We achieved the first neutrons with the system in August of 2021. We also achieved what we considered to be the minimum performance level needed for human clinical use in September of 2021. The proton beam energy was 2.3 MeV and 7 milliamps of current. The stability of the proton beam was very good, better than 1%. So just talking about some of the graphics, the top right graphic shows the stability of the current of the proton beam here running for about 50 minutes in that time slot. Again, very stable. The bottom graphics show how the neutron dose is scaling as the beam energy increases, as the beam current increases, exactly as we expected from a performance standpoint. We are continuing to scale up the beam performance of the accelerator, and we have already commenced doing animal studies with new boron in China in 2021, and those will continue in 2022 as they move towards their regulatory submissions for the Chinese FDA. Here's just some pictures that were taken at various points during the install. I showed you the schematic of the accelerator before. The top image shows the actual installed unit there. These pictures are maybe 6 months old now at this point. Shows kind of the beginning of the beam line and then going through the accelerator and then to the wall and then into the next room, which is a beam line room, which routes the different beams to each of the 3 treatment rooms. So as was discussed in some of the earlier presentations, the accelerator is only part of the technology that you need in order to make a fully capable clinical system. So we are building all of the technology involved in a full BNCT system. Our partnership in New Boron, with New Boron, we're exclusively providing the accelerator to them, but they have their own technology, their own platform for doing the rest of the BNCT treatment. For our installs outside of China, we will develop all these pieces ourselves. So things that were touched on the earlier presentations, the treatment planning capability in partnership with research, the robotic treatment couch in partnership with BEC. We'll have our ICP OES, which will be used for blood boron measurements, much like was described in the other talks. We have an innovative beam shaping assembly that will generate a very good neutron distribution, heavily oriented toward the epithermal energy range, which we think will be very important for clinical use. And then all of the other components that you need to ensure safety and making sure that you're monitoring the dissymmetry of the beam properly. So these are all developments that we anticipate completing in 2023 as we start to install our first full BNCT systems. So having the drugs in place, a key technology for us, key products. We're very proud about where we are on that. Also having the technology platform, the accelerator and the alpha beam product. Those are all key. But you also need, as Dr. Sauerwein said earlier in this discussion, you need a large amount of clinical collaborators in order to do the clinical studies necessary to achieve approval. So we've been in the process of finding those partners around the world. To start first with North America, we signed MOUs with 2 leading medical centers. And we have 2 more MOUs that are in process, that are in our pipeline that we're trying to close. We also have some research collaborations with MD Anderson, some of which will result in collaborations, publications, I should say, in the near future. So stay on the lookout for that. Our goal in the United States is to have 3 to 4 BNCT installations to really get us through the FDA process in the United States. Switching to Europe, we have a signed contract. I was just at this center last week in northern Italy near Milan. This is the team in Pavia. The center name is Canal. They are the only heavy ion system in Italy, a very innovative oncology center there. They have now started the construction process. Groundbreaking just happened there, and they're moving forward to be our first customer in Europe. We also have a signed MOU in the United Kingdom, a very innovative center there. And I'm happy to report just yesterday, so brand new for this meeting, we signed an additional MOU in Europe. And we have one more that we're working on as well. So again, you know, kind of very good progress in Europe. We plan to have 3 to 4 BNCT installations there, which will enable us to achieve approval of our products in Europe. And then switching to Asia, in Asia, our focus has been more of a partnership type model. I mentioned before the installation in Xiamen, China, that will be part of New Boron's BNCT system, of which we are providing them the accelerator for that solution. They call their product NUPEX. There's additional orders in China that New Boron is working on, and I believe will come in over the next year or two. We have a great collaboration with Kyoto University that I mentioned before, and some of the work that I presented earlier will likely end up in a paper that you'll be able to see at some point in the future. So through these partnerships, we think we'll have between 2 and 4 BNCT centers going in Asia as well. So putting it all together on the clinical development side, between North America, Europe, and Asia, we think we'll have 8 to 12 BNCT installations over the next 5 years, some of them direct where we're focused on them, some of them through different partnerships. We also expect multiple academic research collaborations around the world. So just to kind of close out the intro to our company and the update for this meeting, our progress continues as a company. In this meeting, we talked about the newly signed MOU for our research consortium in Europe. We have 2 new drug product candidates since our last public presentation, which was not long ago at ICNCT 2021. So we're making excellent progress there. We've continued to progress on our accelerator installation and commissioning in Asia. And we've now conducted successful BNCT animal test results for our new BNCT drugs at Kyoto. So just really excited to report that progress. And lastly, I want to just talk a little bit about collaboration, something that Dr. Sauerwein focused on earlier, and I couldn't agree more with his sentiments. There's going to be a need for significant industry and clinical collaboration to fully enable BNCT in the clinic. For our company, we're very partnership oriented, and we're open to many collaborative scenarios, both with industry partners, with potential competitors, as well as with clinicians and researchers. And one of the things we would like to do is test some of our new BNCT drugs as broadly as possible. So with accelerators other than the accelerator that we are making, we would very much like to work with Neutron Therapeutics and Sumitomo, the other speakers in this meeting, to test these drugs and bring them to market as rapidly as possible. So if there's any interest, please get in contact with us to talk about those opportunities. So I want to again thank all the speakers for being on time and thank our hosts for having the meeting. Look forward to seeing some questions and answering them in the chat. Thank you very much.
Great. Thanks, Rob. Excellent, really excellent talk. And what I really like is how you tied everything together after 3 hours of learning. A tremendous learning experience for me personally. And now I have the difficult decision to decide, are we going to entertain a couple of questions? There's so many questions out there in the Q&A. And I think, let me ask Vanya's input here. I think it's a long 3 hours. I wonder if it makes sense just to close it and continue the online discussions as we planned. Shall we do that, Vanya?
Yeah, sure. I mean, we're collecting all the questions and we'll get all the answers and send them.
Great. So let's do that. I know there's still 220 people. I saw people, it's late in Japan, it's lunchtime in the US. it's late in Europe. So let's conclude then this excellent session. I cannot say any more that I learned a ton in the last 3 hours. Thank you very much for all the speakers. A huge thank you for Vanya and Anya for putting this together and make it such an incredible, smooth operation. Running for 3 hours with no single glitch is incredible. Thank you very much to Cosylab for the initiative. I started talking to Mia a while ago about the need for this, and I think want to just conclude saying that we are planning a series of these meetings, because I personally started my career in South Africa in 1988 with fast neutron therapy. I know Wolfgang since those days, and I'm really excited to see the tremendous amount of work right now in this space. The key element here is that now BNCT can become hospital-based, and patients doesn't have to travel to a nuclear reactor. And we heard several times through the day difficulties of just the association with nuclear power and nuclear reactors and all that stuff. So I think I personally am really excited about the future and how this can work together with in the hospital setting. I do have my own couple of personal questions about combining this therapy with really advanced proton therapy or carbon ion therapy or even IMRT, and I'll pose some of those questions as we go along. Right, so then in conclusion, we will send out a bit of a survey to everybody, and the question just what would be the topic for the next talk? I mean, I do think the next one we will definitely do shorter than 3 hours, probably maybe a more 45-minute presentation where we can really focus in on specific topics. And I think Rob's presentation tied things very nicely together. And we will ask in the survey, what did you learn? What is it that you want to see next? And continue to do that. I'm also a big supporter or member and participating in the PTCOG environment, and where PTCOG used to stand for protons. It's not protons anymore, it's particles. So everybody in the neutron space is want to align with Jay Flanz's opening remarks that we want to see the BNCT subcommittee grow in stature within the PTCOG environment. And our next PTCOG meeting, of course, is end of June, first day or two in July this year in Miami. So anybody want to participate there is welcome. So with that said, then thanks again to Cosylab for putting it together, Vanya and Anya for the excellent arrangement, and all the speakers. And with that, I would say thank you very much. A wonderful day and a great learning experience. Thanks, everybody.
Thank you.
Thank you very much. It was a great session.
Thank you.